Welcome back to Exploring the Cell with Clay, Rodney, and Cameron. Today, we will be learning about the process of protein synthesis. It is a two-step process beginning with transcription.
As you can see here, DNA consists of two strands of nucleotides. Nucleotides are the subunit of DNA, and they are made of deoxyribose, a nitrogen base, and a group of phosphates. The nitrogen bases can either be adenine, thymine, cytosine, or guanine, and they are held together by hydrogen bonds in the middle.
On the other hand, mRNA is a single strand of nucleotides. It is similar to DNA, but it has ribose instead of deoxyribose. Instead of having thymine as a possible nitrogen base, it has uracil.
Transcription is the process of mRNA copying DNA. An enzyme called RNA polymerase splits the double-stranded DNA in two, creating a template for the mRNA to be created from. A pre-mRNA molecule is then made by matching up the complementary nitrogenous bases from the template to how they would look on the other side of the DNA.
Once pre-mRNA is created, it must undergo a refining process that purifies it into mRNA, as you can see here. Protective molecules are added at the end of the strand, and parts of the sequence which serve no purpose are then cut out. The final product is able to leave the cell and travel to a ribosome, where it can be translated.
Transcription takes place in the nucleus, where the DNA is contained, and it has to take place here since the DNA cannot go outside the nucleus, whereas the mRNA that is created from this process can. Next up, translation.
mRNA leaves the nucleus, where it goes to seek out a ribosome to connect to. Once connected, tRNA arrives at the nucleus, bringing amino acids along with it. The codons on the mRNA, which are a series of 3 bases, attach to their corresponding anticodons of the tRNA.
The process then repeats itself, moving down the mRNA's codons until it is finished. Each new amino acid that is added connects to the previous one, creating a chain of amino acids held together by peptide bonds. This chain is known as polypeptide. The polypeptide then leaves the ribosome and folds into a protein.
We hope you enjoyed this lesson on protein synthesis. See you next week, same time, same place, for a lesson on photosynthesis.